first, I will, in this lecture, first I will speak about space and time scale of biological interest. And then I will give some information about the atomistic and coarse-grained models. And then I will give some details about the two most common approach for coarse-grained model that are classified as top-down and bottom-up approach. So, as most probably we all know, the most common form of DNA is the double helix, which is formed by two chains that wraps around each other in an anti-parallel fashion. And in the most common form of DNA, that is called bDNA, the wrapping of the two chains around each other lead to the formation of two grooves. One is narrower and is called the minor groove, and the other one is larger and is called the major groove. In each cell, we have 23 chromosomes, which means that we have about 3.1 giga base pairs. So in the diploid cell, we have for each cell about 2 meters of DNA. It's an impressive amount of DNA that we have in our body. If we make a calculation of the DNA length on average human body, the length would be enough to go back and forth to the moon many times, 200 times or more. So we have a lot of DNA in our body, and we have a lot of DNA in each cell. And the 2 meters that we have in each cell are very compacted in particles in the nucleus that has a diameter that range between 5 and 20 micrometer. So the DNA is highly compacted and still working in the cell. And at the most fundamental level, we have the double helix that I already described, but and then the double helix wrap around proteins that are called histones, and about 160, 170 base pair wraps around these proteins, 8 proteins called histones that are compacted together and form the so-called nucleosome core particle, NSP. And this nucleosome core particle fold in a more compact structure that is the chromatin. And the chromatin exists basically in two forms, in euchromatin and heterochromatin, that is more condensed, and the heterochromatin and transcriptionally not active, and the form that is less condensed, the euchromatin, and transcriptionally active. So the DNA folding nucleosome core particle, the nucleosome core particle wraps in fibers, and these fiber fold in higher order structure whose features are not completely determined so far, and finally forms in fold in the chromosome. The DNA in the cell is not only in the form of the double helix. In the double helix, we have two bases that form hydrogen bond with each other through the so-called Watson-Crick hydrogen bond. And but the DNA in the cell can organize temporally also in other type of structure. One of the most relevant sees four bases pa- forming hydrogen bond with each other uh, in a different fashion, of course, than in a double helix. These are called Hookston hydrogen bond, and these four bases form a tetrad, and the tetrads can pile on each other, forming the so-called quadruplex structure. And there are many ways in which a single DNA chain can form a quadruplex, and often in the cell or in the samples in the wet laboratories, depending on the particular uh, environment in which the chain is found, high concentration of ionic species, type of ionic species, fall in a different way. And these structures of quadruplex are found either in the terminal part of the DNA chain in the chromosome that are called telomeres, or in a particular region of the DNA in the cell that are biological relevant, like promoters or replication region. And they are considered an ideal target for drugs for inhibiting the growth of cancer cell, or for even in antiviral application. Here is shown a picture taken from a paper of Nature of 2013 that is considered the final demonstration that this type of structure, the quadruplex, actually forms during particular phase of the cycle of the cell. In particular, here these red spots are due to the fluorescence of binders of molecules that bind specifically to the quadruplex DNA, and so it demonstrates that the quadruplex DNA form. Indeed, quadruplex DNA structure were discovered almost at the same time of the double helix, but only so recently it was finally demonstrated their biological role. Let's spend few words on the time scale occurring in the bio world. We have phenomena that involve the molecules in the living organism that occurs on very different time scale. So, for example, the light absorption, which is very important in many processes, some of which can be dangerous, like the modification of the DNA because of the exposure to the sunlight, occur in a very short time scale, below the femtosecond time scale. Other phenomena like proton transfer, which is also a phenomenon of highly relevance in many biological activity, occurs on a on many order of magnitude slower phenomenon or slower scale. And if we think about a phenomenon like protein translation, this process can occur in a much longer time scale. Uh, we can speak about microsecond, second, and even beyond. Uh, so, how do we describe, how do we create a model for the phenomena that are occurring? It depends on the time and on the space scale. During the school, you have learned and heard many talk on phenomena occurring on very different time scale and for which different method has been used. When we work on phenomena that involve the electron variation in the electronic structure, like chemical reaction or catalysis phenomena, we need to use quantum mechanics if we want to study the modification in the electronic structure. We need to go through quantum mechanics. And this also means that we can limit the calculation on very small space scale and short time scale, as indicated in the slide. These are the only methods that allow us to study the reactivity, so the formation and forming of the bond, and to quantify the barriers of the process, and to allow us to identify possible paths that are followed and how modification in the chemical structure can affect them. If we need to study phenomena on a longer time scale and larger space scale, we cannot simulate anymore the system with the resolution of nuclei and electrons, but we need to go to coarser level and consider as particle something called atoms, that is the merging of electrons and nearby nuclei. With this type of scale, we can study the structure, dynamic, and thermodynamic of mixed liquid. We can study solid, we can study interface, both at the equilibrium and at non-equilibrium, because many phenomena of interest, life is at non-equilibrium phenomena. But if we need to increase further the size of our system, and the time scale, as indicated in the slide, we need to have coarser particle than the atoms. And one way to reach coarser particles is to merge together different atoms in a single particle, and to consider aggregate of atoms. So here are shown, for example, hydrocarbon chain, where several CH2 groups are grouped together in a single particle. And in this way, we can study the behavior at an interface for longer time, and larger space scale that we could with atomistic scale. In this with this method, we can study transition phase in ordered system that are difficult to study even at the atomistic scale, and we can sample large conformational space, like formation of micelle, lipid phase variation, and so on. Interaction of DNA with protein in large complexes. And if we need to go to larger scale, to go on time and space dimension that are more related to what our senses perceive, the view, and we need to go to method where we don't even have any more particles. Method like lattice Boltzmann and continuum method are widely used to study hydrodynamical and mechanical properties, and in most cases they have, as is written here, no close connection with the atomic particles. Going to the DNA, that is the subject of this talk, there are many phenomena that, example of phenomena that occur on many different scales. So and therefore can study with different method. So quantum mechanics, for example, is necessary to study the modification of the DNA that occurs through the interaction with the light. And very often, if we want to understand the phenomena, we cannot limit the representation of our model just to the site where the reaction is occurring, but it's important to consider also the fact of the surrounding part. But if the size of the system that we are modeling increase very much, we cannot use any more quantum mechanics. So what is often done is to combine quantum mechanics and molecular mechanics with atomistic description of a part of the system, so that we can have the effect of the surrounding part on the site of reaction, but we can limit ourselves for the part surrounding the active site to a simpler description. And if we are not studying reaction, and we are studying the structure of the DNA, how it is modified by the sequence, by the interaction with ions, by the interaction with drugs that bind not covalently. If we are studying phenomena that do not involve reaction, we can use atomistic scale, or in some cases, if we need to see modification on some on the atomistic scale that are connected to larger transition that we cannot model at the atomistic scale, we can use mixed model of molecular mechanics and coarse-grained model. Sorry. And if we have to study large assemble of DNA, maybe with the protein that form the histones, and how these phases changes depending on the environment or some signal, then we have to go to the coarse-grained level. And only this way we can simulate, for example, the assembling of the nucleosome core particle of which I spoke a few slides ago. The method I mentioned in the previous slide, the combination of two methods like quantum mechanics and molecular mechanics. And this type of combination is called multiscale modeling, multiscale method. There are mainly two family of multiscale method, and one type is the simultaneous or embedded multiscale method. In this type of multiscale method, we simulate, that is the type that I mentioned in the previous slide, we simulate a part of the system at a certain resolution, for example, with quantum mechanical method, and another part of the system at a lower resolution, and we can even have another larger portion of the system simulated at even coarser resolution. And this type of method is called simultaneous embedded multiscale method. But there is also another type of multiscale method that is very important in biological simulation and in simulation of DNA structure. and is the hierarchical type of multiscale method. Um, in this type of in the hierarchical multiscale method, we model the system at a certain level of resolution. So, for example, at the quantum mechanical level, where we have nuclear electron, and from this very high resolution description, we develop a model that retain only certain features of the high resolution model, but reproduce selected properties as in the quantum mechanical method. And from this atomistic level model, we can reproduce, we can use the same hierarchical procedure to obtain a model, a coarser model that reproduce selected properties of the atomistic method. And from a coarse level, we can get even to a coarser level of description of the system, and therefore larger scale, deciding which property we want to model when we are developing coarser and coarser model. So the idea is I start with a very accurate model, and then I remove some descriptors of some details of the model in order to model a larger system. And this is called the hierarchical multiscale method. Concerning the DNA structure, there is a lot of investigation through modeling and through experiment, of course, going on since many decades, and with the collaboration of Sergi, we are somehow trying to contribute in our way to the system. But despite the effort of many scientific groups, there are still many questions that are not fully answered, for which there is not rules that we can apply generally to answer to the question, like how the sequence of the base affect the wrapping of the DNA around the nucleosome, or how the denaturation of one structure of the DNA and formation of another structure of DNA occur, how different ions affect the DNA flexibility and the DNA condensation. There is still a lot of work to do on this phenomena. We have answer for some limited case, but we do not have the general knowledge to know, oh, if I put mercurium, then this will happen with this sequence of DNA. We still have to understand many of these things. How the concentration affect it, or when we have two DNA bases that there is a mismatch in the double helix, how long the structure alteration due to this mismatch will alter along the DNA chain, affecting property like flexibility, or that can have a long range in the space effect, and how the higher ordered structure of DNA organize in the cell. There is continuous knowledge growing on this phenomena, but still there is a lot to do in this field. How the flexibility of the structure is altered, of the DNA is altered when, for example, we go from a double helix to a quadruple helix that I mentioned before. So there are many, many questions that are of relevance. And you have seen in the previous talk, you have heard about many application of atomistic simulation in which you use force field where you have terms that correlate the energy V of the system with forces between atoms, like the so-called bonded interaction, like bone stretching, angle bending, torsional rotation, and non-bonded interaction that occurs between atoms in our system. Um, in coarse-grained model, we have a description of the system that is coarser than the atomistic description in what we call coarse-grained model. And the model is simpler than that of the atomistic model, and it can be obtained basically in two ways. It can be, and these two ways are called top-down model and bottom-up model. In the top-down model, we start from a simpler a concept that we have of the system that we want to model, and we develop a model of it that is simpler of what is the reality of the model. So, for example, we can consider, we know that the DNA is organized in a double helix in most part of the DNA in the cell, and instead of considering that all the atoms wrapping in the two chain, we consider the DNA chain as formed by beads connected one to each other. And we study the flexibility, the folding with this model. So this model is a simplified model that comes from the idea, macroscopic idea that we have of our system. Or we can have a simplified model in which we start from a very detailed description of the system, and we, starting from that very detailed description, we simplify the model, grouping part of the system in coarser system. Let's say that as a general consideration, in the top-down model, we develop a simplified model, and we develop potential of interaction of the particle forming this model by reproducing experimental observable like the melting temperature, the persistent length, the force extension curves, and so on. While in the bottom-up model, what we do is different. We don't try to reproduce the macroscopic properties, but we try to reproduce with our coarse-grained model the properties obtained at a more accurate level of description. And what are these properties that we try to reproduce? This can be structural properties or forces between the particle. So these are the main type of way of obtaining a coarse-grained model, but it should be mentioned that now is becoming more and more common to develop a hybrid model where the two approaches are somehow combined. I will not go too much into the detail in this lecture that is introductive. So, as I said, in the top-down model, an empirical model is built based on some idea of the important features of the model, of the molecule, and the parameters are adjusted to reproduce some experimental data. And I already mentioned the bead and string model for DNA or worm-like chain model. And there are many other models that have been developed in the last decade for DNA, and I will give you a better idea on some very little details on a few of these models, like the Pablo and Aldrich model that are used for DNA hybridization, and the Martini model. So in this top-down model, the Pablo model, there are different, there are sorry, for the bases, there are four different base sites, one for each type of a base in the DNA, so adenine, thymine, guanine, and cytosine, and for the each of these basis, it's developed a free site per nucleotide model. So the atoms, for example, in the phosphate or in the sugar and in the and in the aromatic base moiety are all grouped together. And so from all these atoms that are shown here, we go for each nucleotide to three beads, as described. And in between these beads, we are between these bits are associated potential similarly to what is done in the atomistic model, and these potential are related in this particular model to bonding interaction between these beads that are not anymore atoms, angle interaction, dihedral interaction, and also stacking interaction between the aromatic moieties and other interaction, and the values at the equilibrium are taken from, in this model, from crystallographic structure, and many of the terms are iteratively optimized to reproduce the thermodynamics of the melting of the DNA, which is the separation of the chain, the disruption of the double helix. The shortcoming of this model are in the treatment of the electrostatic interaction through the Debye-Hackel model, and ions are spherical and not polarized by the surrounding electric field. And the solvation of ions is generally ignored, except its effect in determining the effective size of the ions. And the solvent, differently from what occurs in reality, plays no other role that of providing a medium that has a constant permittivity. This model has this limitation, but it has been extremely successful in reproducing very important properties of the DNA, like the thermal and mechanical properties. So here are represented the so-called melting curves of DNA, in which it is shown at varying temperature here in the x-axis, how the structure, let's say, deviate from the original structure, and let's say that the temperature at which there is the disruption of the structure is where we have a flex here in this curve. And these models have been very successful in reproducing this temperature, as it's shown in this graph, where experimental and simulation data are compared. And this model is also been very good in reproducing properties like the persistent length, which is an indication of the flexibility or rigidity, if you want, of the molecule, even as a function of the salt concentration and the mechanism also of hybridization when two chains form a structure. But this model is not addressed, for example, also to reproduce structure deviating from the canonical B-form. And it cannot be used in the original formulation to see the difference in the behavior with different salt, which are known for sure to have very great effect on the structuring of DNA. And lately, anyway, this model has been improved, including some feature concerning the specificity of the interaction with the ions through a bottom-up approach. There are also other models, but I see that I am going very slow today with of the type top-down. So I will skip this one of this model in general, to go and speak about in general the limitation of this top-down model, as part of which I have already emphasized with the first model. And in general, the top-down approach are not capable, because they are not constructed for this reason, to properly treat the specific interaction of the nucleic acid with the ions. That is key of many phenomena related to DNA. In the bottom-up approach, the model, as I said, are systematically developed based on more accurate, more resolved, higher resolution model. So in particular, the coarse-grained model are developed from atomistic simulation, but we can also develop atomistic model from QM simulation. And in general, these these models are based on statistical mechanics. And we can group this bottom-up coarse-grained model in two big family. One family is the development of coarse-grained model that reproduce structural properties, and one family that is reproduce forces. And I will go to the description of the force of the coarse-grained model developed to reproduce structural properties, using as method of reference a method that has been developed by our co-worker Professor Laxonen and Professor Liu Bartsev, Sasha and Atto, more than 30 years ago, and is one of the first model, coarse-grained model for DNA of the type bottom-up. Mathematically, we start from a function that is function of many variables, and we decide that we want to simplify and describe the system with less variable, and we approximate the problem of an n-body potential that is not mathematically amenable with a sum of two-body potential, and then we choose the form of the two-body potential. So in the method developed by Lyubartsev and Laksonen, it's based on something called radial distribution function, that are the function that describe the average distance, the distances that the particles have on average from each other, and the probability to have for each distance that the two particles are at that distance. And from this function, through a method called Boltzmann inversion, from this radial distribution function that we call G of R, we develop potential, and we optimize with an iterative scheme this potential until we have with this potential, with the coarse-grained particle, the same spatial description that we have with the atomistic description. And now I will go a bit quick. I will speak some slide. Today I went really slow, showing to you the scheme, the idea of this coarse-grained methodology. So basically, we start with an atomistic simulation where we have, for example, a fragment of DNA, the ions, and the solvent. And then we decide which particle we need in our description of the method. What is the simplification that we want to make in this method? And in this case, we want to describe the double helix of DNA and keep only this particle, but still, so we decide how we group the atoms in beads, and we can decide to group as we want. So for example, as we have seen in the top-down method, that we have one bead for the sugar, one bead for the phosphate, one bead for the nucleotide, we can group. Okay. And then in this step, we are removing in the description the solvent, but not its effect, because we decide how these particles interact with each other, and we decide which spatial distance we want to consider. So for example, the distance between two phosphate or two bases that are on opposite chain, and we calculate and the atomistic, from the atomistic simulation, the radial distribution function, so the probability of finding at a certain separation these particles. And from this radial distribution function, through the process of Boltzmann inversion, we obtain a potential. We use this potential to simulate the system with, for example, molecular dynamic simulation or other method like Monte Carlo simulation. So we use this potential to model the coarse-grained system, and from the coarse-grained system we calculate the radial distribution function. And if the radial distribution function do not coincide with those obtained atomistically, we optimize the potential till the coarse-grained and the atomistic description of the system do not coincide. And they coincide, it means that if we are not using the solvent in our description, in our simulation, so we are simulating much less system, we have the effect of the solvent in our structures because we are reproducing radial distribution function that take into account the effect of the solvent. And this method has been applied to simulate the interaction with different ions. So, for example, different alkaline ions, and is able to reproduce differences when the ions are different, and also to reproduce a property like flexibility at different salt concentration and with different ions. And and The method can be used for, has been used by Sasha Lubartsev and co-worker, that is one of the developer, also to go from atomistic simulation to coarse-grained simulation, and from coarse-grained simulation to coarser simulation. So going from an atomistic description of the nucleosome core particle to a coarse grain and even coarser grain. So we go from thousands of atoms to, for example, in this method, in this model, 7 particles. And then you can, once you have simplified so much your system, you can see how the nucleosome core particle group together and make hypotheses of how the DNA is really packaged in the cell. And these hypotheses are informed by atomistic simulation. So I will not go into the details, but this method can be applied for different type of structure of DNA, and we have applied it to also to quadruplex of DNA to study how these quadruplex of DNA group together in larger structure and how the physical property like flexibility change. And the simplification of the system are huge. You go from a system where you have just for the DNA, for example, 800 atoms, but for the echo simulation, thousand and thousand of atoms, to system where you have for a quadruplex only 21 bits. And my final note is about the limit of his method. So you start from atomistic simulation, and in atomistic simulation you have a lot of different set of parameters that you can use for your ions, for the water, for the DNA. And if this simulation, atomistic simulation, have defect, this defect will be used, will be transmitted to the coarse-grained model that is trained to repeat the same simulation. So now I will skip the slides, and I will, my final remarks to leave space to some question is that the particular model that is used at the coarse grain level to study any system depends on the property you are interested in. Because anytime you increase the level of approximation, the application of the model decreases. So you decide what information you want to have, and you cannot use the model to obtain information for which the model is not trained. And in bottom-up model, any limitation of the method, the description at the more resolved level that you have is retained in the coarse model. And also, this is a field that is in great expansion. So you now there are many hybrid model, and before you decide which model to use, you need to consult the literature concerning similar system. And finally, I want to thank you for the attention. Thanks and express my encouragement and gratitude to our Ukrainian colleagues and friends, whom, despite the difficulties they are having since now many years, they are continuing strong in resisting in this terrible war and still being able of doing great science. Thank you.
Thank you very much. Thank you very much for such wonderful lecture. And now we have some time for questions. So please ask in here, maybe in room, in Zoom. Maybe people are thinking. I just ask some questions that I suppose maybe on the surface, especially after our previous lecture that was about neural network. So is it possible to use some methods like machine learning or neural network together to develop such coarse-grained models? Maybe this technique is already used.
I think the more is periodic and organized the system you're studying, the more this type of method can be used. And I think they are already used in solid state and, as I said, more periodic system. Even if I am not aware of any uh, of any coarse grain model for this system developed in this way, I think that in the future we will have. I mean, it's obvious that this method, if properly trained, can find uh, um, correlation that are what is the basis of the interaction potential that we have in our model. So I think we will see this in the future,
Yes.
even for biological system. I see there are now, I was surprised to see for ionic liquid, that is another part of my interest in science. I'm interested, let's say, in general, in highly charged system. Surprisingly, that are a matter that is not periodically organized. It's liquid. But I saw that lately there have been atomistic potential developed with machine learning. So I think that also sooner or later, we will see the coarse grain potential also developed in this way.
Thank you very much. We have one question. Please start.
Okay, Francesca, thank you very much for so detailed and nice talk. So my question is very basic, and I would like to know a bit more about the program packages that I can use to perform some coarse-grained simulation. For example, for the, I don't know, DNA origami or something like this. Thank you.
Thank you, Tatiana, for your words and for the question. And so this allow me to give me the occasion to show the link to the page of the program developed in Stockholm. There are so it's a completely free program. Is one of the many program. Is the one that I have been using in my group. We have been using, and it's, I have to say, it's free, but it's not easy. I mean, it's not free in time that you will use, because it requires extensive atomistic simulation, and then it's you have to develop for each system that you work a model and think about which particle will interact with another particle. So it's and you do this only when you have a system where you cannot have with a simpler model, simpler to use, like Martini force field. Good answer. And well, this is often the case of interaction with ions, which is something that interests you too. And the step of, I am very sorry, but in this period, I was not even sure I would have managed to give the talk because of the car, but luckily, yes. And also another part that is very long is the convergence of the potential. So this part that I described here, so you obtain the radial distribution function, for example, from the atomistic simulation, and then you have to converge the potential from the initial to initial distribution to the final distribution. This can take time. There are also other, I don't have here the slide, but I can provide you also the indication for other program, but for this Stockholm program, this is the link. The program is called Magic.
Thank you very much, Francesca, and also maybe other questions. Yes, Professor Slissonen.
Thank you very much, Francesca. I'm very beginner with this coarse-grained model. So for the dynamics, do you perform Newtonian dynamics? What is the time step, and how long are the simulations?
Another very good question. So we the time step that we use even at the coarse-grained level is not so much longer than the time step that we used in the atomistic simulation. So we are still in the femto second time scale. And how long it is the simulation, it depends, but we are typically in the system like the DNA, the quadruplex, and in the microsecond time scale. So it's still computationally intensive, but we can reach scale that atomistically, the scale in size that in the atomistic time scale we cannot reach.
Yes,
More than in the time with this method, we increase in the space, let's say.
So due to the reduction of the number of particles, we increase the time scale trajectory. This is okay. Maybe other questions. Maybe questions in Zoom. We have several questions in our room. Maybe in Zoom somebody. Also, maybe in chat. Please.